So next slide. So as I said, we're at the Lynbrook chapter of the IYNA, which is these are all our pictures. Steve is running a little late right now. He'll join our meeting later. I'm Sayem, and
I'm Anisha. I'm the vice president.
all right. So basically, we're all a bunch of high schoolers, college students that have a bunch of members around the world, and we just want to share neuroscience to our community and to anyone who finds it enjoyable and fun. All right, and then we're just going to skip these slides. We're going to get straight to the dissection. Neuroscience is just the nervous system, what our brain is, structure of the brain, etc. Let's get right into it. Okay, so before we begin, we need safety, right? So I'm going to put on some gloves and some goggles. And then this is a sheep brain dissection. And so as you can see, that sheep brain is kind of, it's more flat. It's not like a human brain, which is on the right. And we bought the sheep brain from carolina.com, Carolina Biological, something like that. And if you want to buy sheep brain too, then you can just go to that site, you put into Google, and then you'll get the brain. All right, so now I'm gonna mute myself. And Anisha, can you go ahead and stop the screen share now?
Yeah.
Okay, so now you should be seeing the sheep brain.
Ooh.
This is the sheep brain here.
Yeah.
Can you, Anisha, can you pin the video to make it so that everyone's only going to be looking at the brain and not at other speakers?
Yummy. What's the scope of share?
Why yummy?
Is it pinned for everyone? It's pinned for me. I don't.
Yeah. I'm looking. Okay, so how about I think everyone just needs to mute themselves. That's probably going to be the best way to do this. Okay, and then Anisha, if you just look at if you can just look at the chat, then like answer a few questions that pop up there, and I'm going to continue with the dissection. I see one question: do you kill the sheep to get the brain? And I think the answer to that is obvious. You can't have the brain without the sheep still alive. So, but sometimes it's like if you're harvesting the sheep for food, then you're not going to do anything with the brain. So, or if the sheep, it's probably not a natural death. Okay, so this is the brain. We can look at all sides of this. So what you're looking at here is the brainstem. And on this side, we have the two hemispheres. And what you'll notice is that, I don't know if you guys have seen a human brain before, but in a sheep brain, the brainstem is actually going horizontal, whereas in a human brain, it would be going down like this. And that's because when you think about a sheep, they're four-legged animals that, and their heads don't curve up at the end like we do. We walk on two legs, right? So this brainstem would continue and become the spinal cord here. All right, so first, there's a lot of questions in chat, Nish, so just make sure you're going through all of those. So before we begin, right, there's a couple of key terms that we need to know. The first is dorsal, which means the back, right? And so the dorsal side of this sheep brain would be the side you're looking at right now, because that's the side facing the back. And then on the opposite side, we have the ventral side, and that's the side you're looking at now, because this is the side that would face the front in an alive sheep brain. So in this brain, there's no protective covering of the brain, right? In our brains, we have layers of meninges, right, that protect our brain from additional damage. But in this brain, since it's specifically for dissection purposes, and it's been soaked in formaldehyde and make sure it's not going to like corrode on its way here, the meninges have already been removed. And so we're looking directly at the brain with no covering. Okay, and so now we're going to cut the brain in half, and we're just going to start at the very top and cut all the way down to the brainstem. We're just going to go ahead and do that. Oops, there we go. try my best to stay perfectly in the middle. Because there's a lot of structures in the middle that we can't see without cutting the brain. So, all right. There we go. As you can tell, I'm not a dissection expert, not a surgeon, but I am trying my best to make sure that this gets cut all the way. And if you'll notice, I'm not holding this scalpel like a knife, right? Whenever you dissect, you kind of want to hold it like it's a pencil. That gives you the best control of the tool. All right. Okay, so now we have two halves. We have the right hemisphere, we have the left hemisphere, right? Okay, so we're gonna just take a look at the right hemisphere right now. We'll put the left away and we'll do some other stuff with it later. But right now, what I just cut is known as the central longitudinal fissure, right? So when the two brains were together, you'll notice that there is like kind of like a dent in the middle. And that's because the brain is split into two halves, right? And so where it's split, that's known as the fissure. And then here, you'll notice that there are a bunch of like dark areas. That's actually where the brain is folding. It's going in and it's going out. But since the brain has been preserved for so long, we can't actually pull apart these folds. Like they're pretty much glued together. And that's because of the solution it's been like preserved in for so long. So this area right here is known as the cerebrum. And that's kind of like where all the gray matter of the brain is. The gray matter is the cell bodies of neurons. And we'll have a lesson about neurons later in the year. But neurons are basically the brain cells that are responsible for thinking, sending signals across the body, pretty much everything. Okay. I'm gonna check the chat really quick. And see if there are any questions. Okay. All right, and we're gonna continue. All right, so the next structure we want to identify is right here. You can see how this area right here has kind of a gap, right? Can you all see this gap that I'm kind of stretching right here? So this is what's known as a, actually, I want to ask you guys a question in the chat. Does anyone know why this gap exists? Why is there a part of the brain that just does not exist? Wouldn't it make sense to have neurons in there or something that would make the brain smarter? Yeah, could you type that in the chat instead of speaking out loud? Cushioning is one response from Andrew Kwok. I'm sorry if I didn't pronounce your name correctly. Do we have any other guesses? I'm just going to say right now, cushioning is a correct answer. There are other answers as well. All right, okay, so that's our one answer. Yeah, so cushioning is a correct answer. The gap right here is called a ventricle, and it's the space that we have cerebrospinal fluid in. So if you've ever heard of CSF, cerebrospinal fluid, it's produced by basically little glands in the brain and also in the spine that provide cushioning, filtration, nutrients to all the cells, not just neurons. There's also other cells in the brain called glia, and it provides everything that they need to live. And it also serves as a physical barrier for injury. For example, if this sheep were to bump its head, without the ventricles, everything would just squish, right? But then this provides an additional layer that prevents it from being injured. Okay, so now we're going to look at the 4 lobes of the brain. And the first lobe is the frontal lobe, right? So that's pretty much this entire area right here. Kind of stops short, kind of about right there. And that's when it switches over to the, that's when it switches over to the parietal lobe. So we got frontal, parietal, and then right at the very end, we got the occipital lobe. And so the frontal lobe is where we think about like thinking, emotions, a bunch of like those higher order functions, not just living, right? So in a human, you'd see that the frontal lobe is really big compared to like a sheep brain, but sheeps are not as intelligent as us. And so their frontal lobes are not that big. And then the parietal lobe is a bunch of different functions ranging from sensing to visual interpretation, right? And then in the occipital lobe, we have most of the visual stuff. We have the visual cortex. We have the visual like integration cortex where the information is transformed into something else. And then on the side here, we're going to have the temporal lobe. And the temporal lobe has a bunch of different functions, and it's quite complex. And there's two important areas that we'll talk about later in the temporal lobe called the Wernicke's area and the Broca's area. And then we'll get to the back half of the brain soon, but there's something we have to talk about in the front half, which is right here. You'll notice that there's these little like things that have been cut off. Do you see this right here? That's been cut. And then right here too, like this little thing right here. So in this brain, the olfactory bulbs have been removed. And so what is an olfactory bulb? Does anyone know? Let me give you a hint. Sheeps have very good noses. They have a very good sense of smell. And so why would they need a really big olfactory bulb? That just means they have a lot of neurons. Yeah, perfect. We got a response from we got a lot of responses actually. Yep, smell, great. The olfactory bulb is responsible for a very good sense of smell in the sheep brain. Sadly, we can't see it here. It's gone, along with the pineal gland, which would have been hanging down. It would have been hanging down kind of like this, just a little circle right there. That's also been cut out, so we can't see that. All right. So now we're going to cut right here, separating the brainstem right here from the non-brainstem area of the brain. So we're just going to cut pretty quick, right? And so now we have, you can kind of see how there's two areas, right? We got one cerebellum, which is the circular structure right here. And you can kind of identify that because I'm not sure how good the quality is, but you can kind of make out how there's a little white area and then there's gray areas. So the question one is, why are the brains so small? And when you think about how big a sheep is, its head is small, right? So you can't fit a big brain in a small head. That's why the brain is small. A bird brain would be smaller, an elephant brain would be huge. That's just how it is. Yeah, so the cerebellum has kind of like this tree of life, right? In Latin, that's known as arbor vitae. And so you can identify that this is a cerebellum. Even if you've cut it away and you have no idea, like if you pick up this piece, if you can't see that tree of life, you know it's not the cerebellum. And so this is the cerebellum. We're just going to cut that out of the brainstem. It's not a part of the brainstem, just to be clear. It's another lobe of the brain. So we're going to go right here, and we're going to cut the cerebellum off the brainstem. And there we go. So now we have one part of the brain, another part that's the cerebellum, and we have the brainstem right here. Not in the exact, not as good as it was before, but that's just how it is. And so why do we dissect? We dissect to learn more by actually physically interacting with things, right? So when we dissect this brain, we don't want to just cut it. Like I know my freshman year when I did a dissection, I just cut it because I wanted to cut it. I just wanted to cut the brain and see how small pieces I could get, right? But that's not what we want to do. We want to dissect the brain so we can identify each individual part and learn more, right? So now we have the brainstem. This is one half of the brainstem. Obviously, the other half is right there. So there's three parts of the brainstem that are important. The first is the medulla, right? So the medulla is the uppermost area of the brain. Oh, sorry. The pons is the uppermost area of the brain. The medulla is the middle area right here. And then we have the brainstem that leads to the spinal cord, right? So, and then the pons and the medulla have really important respiratory and cardiovascular properties. In the pons, we have something known as the pontine respiratory group. And that's why if someone is shot in the arm, sorry, this is a kind of morbid example. If someone suffers injury to the arm, they'll most likely be fine. But if someone suffers an injury to the spinal cord or the brainstem or the brain, that causes death very quickly because we lose our control over breathing. And I say control, most of these groups are responsible for involuntary control of our breathing, heart rate, etc. Okay. So now we're going to look at, we're going to go back. We're going to look at the gaps right here, the ventricles. And what we're going to look at is the area right above the ventricle, right? So all of this right here, all of this right here, we're just going to cut out this small little section. There we go. Perfect. And you'll notice how this small area kind of has like that a streaky pattern to it. And it's really white compared to the gray area that here. So if this was gray matter, does anyone have a guess as to what this white portion of the brain is?
White matter?
Yeah, perfect. That's the answer right there. So this is gray matter, this is white matter. And the gray matter is, Anisha, could you possibly mute? Okay, it's fine, the video's back. So yeah, this is gray matter, this is white matter, right? So the gray matter is the cell bodies. And in a neuron, we have the cell body, and then we have different parts called axons. And so, yeah, Anisha, there might be a setting which might make it so you can't unmute people. I'd prefer if you guys just use the chat because unless this video is pinned, if someone else talks, then the video goes away, right? We don't want that. So if maybe if the video is pinned, we could have other people talking, but for now, we don't want that. Okay, so what was I saying? Neuron, cell body. Okay, so the cell body is the gray matter, and the axons are the white matter. And the reason why it's so streaky is because for each cell body, let's say we got like a circular cell body like this, we have axons that run really, really far to connect to other neurons, other muscles. So for example, if we have an axon in our spinal cord, in our lumbar region, which is the very low part of the spinal cord, that neuron with the cell body in the spinal cord has to run all the way down to our toe so that our toes have feelings, right? And what's running down? Not the cell body, the axon, the one that has a really, really long length. Okay, so why is this streaky? Because all of this is axons, right? And so when we put it back together, right here, I'm gonna put it back, I'm gonna connect the brains together. You'll notice that when I close it, all the white matter is in the inside. That's because the neurons from the right part of the brain talk to the neurons from the left part of the brain via their axons, via the white matter. And that's why this is so important. In epilepsy patients, unfortunately, in past years, like 1970s, 1980s-ish, since we didn't have like drugs that would prevent seizures and stuff, what did help was actually splitting the brain in half, cutting all of these white matter connections. And that actually helped reduce epilepsy. But it also made it so that, for example, you would have a person who could move or could get information from their left side, but then couldn't act on it, right? So maybe if they saw a ball flying towards their head from the left side, that would be information that's passed to the right side of their brain, right? Because you know how information crosses over. And vision's a little different, but the idea is still the same. They couldn't use the left side of their brain to move the right side of their body, right? Could you imagine how that would feel to just have like one half of your body only being able to act on impulses? So yeah, it did help, but it had severe drawbacks and consequences. All right, so now we're going to continue our dissection. I have a little guide here to help me. So now we have pretty much gotten the entire outside of the body, right? The outside of the brain. We got the frontal lobe, we got the parietal lobe, we got the occipital lobe back here, we got the temporal lobe. So I'm going to elaborate a little bit more on the two areas I mentioned before, the Broca's area and the Wernicke's area. So in a sheep, these areas do not exist. Does anyone know why these areas do not exist? I'm going to give you a second to answer that question in the chat. Sandy says they don't need it. Anyone else? Yeah, that's right. The question was, why do the Wernicke's area and the Broca's area, which I will explain what they are, why don't they exist in the brain, in the sheep brain? Yeah, but Sandy got it. They don't need these areas. So the Broca's area is responsible for speech production, and the Wernicke's area is responsible for speech comprehension. And so one like cool mnemonic that I learned was that Barack Obama is a really good speaker. So the Broca's area, which sounds similar to Barack, is the speech production area. Right, so as a human, we need speech. As a sheep, you don't need speech. So these areas don't exist in the sheep brain. Okay, so now, right here, since I've cut out the axons, sorry, I didn't give you the specific name for them. Yeah, the sheep can communicate. Yes, they can say ba, but they don't have an advanced area of the brain devoted to speech production. They probably have lesser areas. So yeah. Okay, so as you can see, I've cut out a lot of this area, which has revealed this small portion right here, right? And so it kind of looks like a circle right here, all of this. And so this area of the brain is known as the thalamus. And so it's one of the most important processing centers in your brain. I know I said that like the frontal lobe is higher order thinking, all of that, but this is a different kind of processing. This is, we get a nerve sensation from our finger. It goes up through the brainstem, up through the medulla, up through the pons, all the way to the thalamus. And then the thalamus chooses where to send it. It could send it first to the visual cortex, or first to the motor cortex, or to this. The signal goes to where it's supposed to go because the thalamus exists. So the thalamus is basically the primary like sensory cortex, all of that. Okay, so now I'm going to remove the cerebellum once again. Sorry, the scalpel is not as sharp anymore. Okay, and now as you can see here, there's more gaps, right? So where we had gaps before, that was the first ventricle on, so the first and second ventricle on the right and left side. Then we have the third ventricle and we have the fourth ventricle. And so near the cerebellum, on this brain, it's kind of hard to see. It's actually kind of impossible to see, but near the cerebellum is a hypothalamus. And I don't know where it is on this brain, unfortunately. It should be right. It should be right around here, but it's not. But the hypothalamus is responsible for like, and maybe if you heard of hypnosis, right? Similar to hypo, because those are both Greek roots, right? And so the hypothalamus is under the thalamus. Oh, that makes sense. Hypothalamus under the thalamus, right? And that's basically responsible for emotions and memory. Okay, and now, yeah, I think that's pretty much it for a dissection. I don't think I'm missing other areas. Oh, yes, there's one more area. Okay, so back to the brainstem. Sorry, I didn't mention this. We got the pons, we got the medulla, and we got the spinal cord, but then there's all the area with the thalamus, with the hypothalamus, with the hippocampus, all of that right here is known as the midbrain. Kind of like trace it out for you right here, right? And as you'll see in our like human brain slides, you'll see how the midbrain is, while the brain develops as an embryo, there, we got like the forebrain, we got the midbrain, we got the hindbrain, and that midbrain develops into what's the adult midbrain. So yeah, does anyone have any questions? Does anyone want me to dissect other areas of the brain? There's some fairly advanced areas here. I can point them out to you on this brain. The folding. Yeah, so the dura mater is also known as the meninges, right? I'm not sure if you were here at the beginning of the dissection, but I mentioned this was a sheep brain made for dissection, right? And so the meninges were already removed. And so there's 3 layers of meninges known as the dura mater, the arachnoid mater, and the pia mater, and they've all been cut away prior to this dissection. If you guys want, we could go into some more advanced areas. This fold, where the corpus callosum kind of folds on itself, is known as the septum pellucidum. That's also Latin. What does the brain feel like? It's like fairly slimy, but I have gloves on and I can still kind of feel its slime. It's not very squishy. It's not very squishy at all, actually. I would compare it to like a harder version of Jello. So yeah, if we had this meeting during a non-pandemic time, we would actually have brains for you guys to dissect, but we can't do that, unfortunately. I'm not sure if I talked about sulci and gyruses, but as you can see right here, these black spots where I'm gonna cut. Let's keep an eye on the time. Yeah, so we might have to end the meeting soon because Zoom only allows us 40-minute meetings, right? So these cuts right here, the sulci are the grooves and the gyruses are like the expanded areas. What part of the brain is responsible for feeling pain and stuff like that? Well, feeling pain is not actually, well, it's part of the brain, but it's a lot more complicated than that. Everywhere in your body, you have, the meeting got upgraded. Okay, we'll see how that goes. If the meeting gets disconnected and you guys want to go longer, I'll just restart the meeting and then you guys can rejoin. So if it gets randomly disconnected, just rejoin the meeting. I'm just gonna keep on going as long as you want to go. I expected 30 minutes, but obviously we've gone past that. So sorry about that. So what was I saying? The brain. So we have receptors known as nociceptors, and they're all over. Like right here, if I stab myself with that scalpel, which would not be fun, I have nociceptors in my hand, and it would go all the way up to the thalamus, and the thalamus would decide what to do with that information, right? So it would either go to like a somatosensory cortex, and then, but the idea of pain is purely in your brain, and there's no specific part of it that's responsible for only feeling pain. What is specific to pain though is the amygdala, which is near the hypothalamus, near the hippocampus, like kind of in this region. I'm pretty sure it's too small to see like in a dissection, but the amygdala is responsible for making sure you don't feel that pain again, right? So let's say, here's an example. One of my friends was riding a scooter, you know, the kind where you like hold it and then you scoot, right? And he rode it really fast and he scooted into a car and then he chipped his front tooth, right? So now his amygdala is going to make sure he does not scoot into a car again. It basically makes it so it takes the pain signal, it realizes what happened in combination with like your motor cortex, frontal processing in the frontal lobe, spatial awareness in the, oh, I forgot to tell you the cerebellum, let's get to that. So yeah, the amygdala combines all that information and tells your brain, not to do that action that caused you harm. So yeah, back to the cerebellum, right? Cerebellum, where'd it go? Cerebellum, tree of life, arbor vitae. Which part of the brain makes you do reflexes? Okay, I'll get to that. The cerebellum is responsible for motor coordination, right? That's why there's so much gray matter and white matter in combination. It makes it so that all your muscles are in sync, right? So because I have my cerebellum in my brain, I can do this, squeeze the brain. I can do this, hold the brain. And so if the cerebellum is damaged, it's really hard for people with damaged cerebellums they have like janky movements, right? They'll like jerk around a lot, and they won't be able to have like a smooth, controlled, like draw a line with the pencil. They can't do that. Okay, so reflexes, like everyone knows the most common one, right? Like a knee jerk reflex, you tap your knee, your leg kicks out. So reflexes actually don't go up to the brain. The brain has nothing to do with reflexes. The reflex is a spinal cord thing. So remember I was talking about how neurons have those long axons? What happens is in your knee, the patellar tendon organ has a neuron connecting to your spinal cord. It sends a signal to your spinal cord. It immediately sends it back to you that says, oh, this has happened, kick your knee out. And then once you kick your knee out, that same neuron that sent something to the spinal cord is still going up. And then you realize later that you've kicked your knee before you've kicked your knee. So hope that answered your question. What part of the brain tells you if you're hungry or thirsty? I'm not actually sure. I'm looking at this right here. The hypothalamus is responsible for internal homeostasis. So I would guess, I'm not 100% sure, but I would guess the hypothalamus or the thalamus, the midbrain, definitely something in the midbrain or this would be responsible for hunger or thirst, but I'm not 100% sure. I could Google that, answer that at the next meeting. White matter, ventricles, medulla, olfactory bulbs, cortex. Oh yes, cortex. So the gray matter right here, you notice how it ends right around here, right around there. It goes from gray matter. I'm not sure if you can see it in the video, but it goes from gray matter to a lot of white matter, right? And that area where it's only gray matter, like right around there, like that just chunk of only gray matter, that's known as the cortex. Yeah, thank you, Anisha, for answering that question. That's good. The next meeting, we haven't decided a date yet. It's always going to be on Zoom, tinyurl.com/iyna Zoom. Anisha, could you start rolling the end of the meeting slides, and I'm going to put the brain away. Or I'm just going to leave it here if you guys want to dissect more, but we need to get the end of the meeting links up for that. The next meeting, if you join our email list, we would be able to send you an email. That's why we needed you to fill out that question form so that we know how you're learning about our meetings. And the link is the same. Yeah. So if anyone has any other questions, I can keep on dissecting. Otherwise, Anisha, do you want to start screen sharing the meeting presentation? And I'll end. I will email you the next meeting date. I'm pretty sure you can still hear me, right? Even though it's being screen shared. Yeah, I will. We will. If you are on the email list that we have, I will email you. So I think we talked a lot about this. There's development, forebrain, midbrain, hindbrain. The hindbrain turns into the brainstem area. Midbrain goes to the midbrain. It's all color coded. Forebrain goes to the cerebrum. Diencephalon is just an extension of the midbrain. And yeah, that's pretty much it. Feel free to unmute yourselves now, though, since the dissection is over. Anisha, you can give them those privileges again. But yeah, at our website, you can find out more. That's going to be updated as soon as we have an, excuse me. That's going to be updated as soon as we decide as an officer team when our next meeting is. Feedback form is available if you have any questions, if you have any ideas, comments, anything you want to tell us to make our meetings better for you, better for us, better all around. Please sign up. If you sign up, that tells IYNA that our chapter exists, and then they can, I don't know what they're going to do, but most importantly, it tells us that you want to join our meetings, and it gives you our email, and that's the most important part. Yeah. Does anyone have any other questions? Otherwise, we're going to leave the slide up for a few minutes, and then we're going to end the meeting. If you do want to buy the brain, it was $15 for shipping as well, so $30 total. Slides are available at tinyurl.com/iwayne slides. That's also on our webpage. So yeah, if you want to buy this brain, it was sheep brain, about $30 on Carolina.